So next we'll have Jeremy Jenkins, who is the US head discovery science at Novartis.
Great. Thanks very much for the invitation of someone from pharma to join this Open Innovation Conference. I mean, we're in the business of trying to make medicines and help patients, not productize AI algorithms. So hopefully I can share with you how we're thinking about using AI in the drug discovery process, particularly generative AI, which is sort of a new flavor, which makes for some very cool research. Historically, we've used more discriminative machine learning. For over 20 years, ML has been a big part of how we do research, particularly in designing compounds using structure-activity relationships, for example. So you want to do something like predict a label or a class given the features of a molecule or input data. So will this molecule bind to this target might be a discriminative machine learning question. But more recently, approaches are available to map the probability distribution of data. In a generative sense, you can then sample from that probability distribution and then use it to make suggestions of, say, what compound to make, what target to test. So this is kind of the revolution of the past several years. And these are just some trivial examples. So, you know, in language prediction, we've all seen ChatGPT. So largely it's training a model on the entire internet and learning how words attend to other words in sentences and in paragraphs, and then it can do things like sentence completion given a text prompt. So that's what most people think of as Gen AI, but generative AI actually touches a lot of what we do in different feature spaces, one of which, for example, is just image-to-image translation. Given this bright field microscopy image, what would it look like if it were stained with an antibody, for example? So how can we pivot from between images? That's an example of generative imaging. We can do this in chemistry. We can sample from a latent chemical space. Say we have a molecule of interest, and we could look in that latent space at what's nearby and then decode it back to a chemical structure. And this is an example here of going from one molecule of interest all the way to another molecule of interest and then sampling the latent space in between. And that's sort of an interesting way to generate new ideas because you're hybridizing molecules in that latent space. So there's just so many applications. On the right is things I'm not going to talk about where Gen AI is impacting drug discovery. So I've just picked out 3 areas to talk about. One is close to my heart, it's target identification. How do we go about picking those drug targets? If we get that wrong, the next several years will be a massive waste of resources. So is there anything Gen AI can do to help? The second is in designing small molecules. You can also use it to design biologics, but I'll talk a bit about generative chemistry. And then third is sort of how can AI help us with our biological assays, enabling them, particularly in imaging where it's very powerful. So how do we get these targets? We heard a little bit about target-based drug discovery, phenotypic-based drug discovery. We do a lot of phenotypic screening, so that is a major source of targets. So you can get targets through small molecules that way, or we can use more of an evidence-based approaches. And classically, you may have tissue abundance of something in a disease setting. We can do functional genomic screening. Was RNAi, now it's more CRISPR, but we can screen for phenotypes and find targets that way. This is becoming almost the classical approach. And then more recently, as we begin to sequence humans across populations and create biobanks, we can do large-scale GWAS studies across different phenotypes and diseases. And those GWAS hits can be pointers to potential causal targets. And we know that targets with genetic backing are more likely to succeed downstream. And then there is sort of just prior knowledge, and there's a huge amount of prior biomedical knowledge, and there is a role there, I think, for large language models to help really summarize and effectively survey it and bring that quickly to a user. And then finally, there's this concept of in silico perturbations. So now we're talking about, can we actually simulate what would happen if we knock down or knock up a gene? What would be the effect on the rest of the genome if we did that? So I'm going to start with that one, in silico perturbations, because it does involve generative foundation models. And I'll just start with an analogy. So if you were to be shown this photo of a party and asked what's unusual about the interactions of these people, your first thought might be, well, it's kind of hard to say. I don't know who these people are, who they normally associate with. So that would be just thinking about this in a vacuum. But what if we had data from 100 million previous parties with these same people? Well, that's interesting, because then we could think about building a pre-trained model on all those other parties and then maybe fine-tuning it on this party to say what's gone wrong. So that's kind of an analogy to what you can do with single-cell RNA-seq data. The reason is there's so much of it now. There's, in the single-cell atlas, we're sort of north of 100 million single-cell transcriptomes across 39 different organs, and this collection of single-cell transcriptomes can be applied to a pre-trained model very similar to what you would do with a large language model. With an LLM, basically you're learning how words attend to other words and sentences. In this case, it's genes are those words and cells are the sentences. So we can train a very similar type of model architecture to understand how genes attend to other genes across cell types. And this is just purely pre-trained, and then we can apply it to any disease setting of interest where you have single-cell RNA-seq data and essentially fine-tune the pre-trained model to learn disease versus healthy. And you can see these models are pretty good at learning what is the difference between disease and healthy cells. And there's a lot of downstream tasks with the foundation model that can be done. One example is just unsupervised clustering of cells. And you'll see that these models are capable of basically distinguishing different cell types from one another, even if they weren't trained to learn cell types, which is pretty interesting. Those ways genes are attending to one another can be used to generate a new kind of gene regulatory network that's inferred from the embeddings. And then finally, that sets us up for in silico perturbations, where we can think about, okay, if I were to take these gene embeddings and pluck out a gene, either knock it up, increase its rank order, or knock it down, what would be the effect on the other gene embeddings? And would that actually reflect reality when we do perturbation experiments? This isn't my idea. There's a lot of work on this in the last 1 to 2 years around foundation models in biology. I've just highlighted some of the ones in green here that can be used for in silico perturbation. We've played with GeneFormer and SCGPT and have invented our own as well. And if you really get interested in this topic, this is a pretty good review article from Fabian Theus on transformers in single-cell omics. So this is all fairly recent, but happening quickly. I'll just show a couple of examples from authors that have used these. So Christina Theodoros wrote the GeneFormer algorithm, which came out last year, I believe, and essentially, she looked at if you knocked down these transcription factors GATA4 and TBX5, would they perturb the embeddings of genes that are actually the target genes of those transcription factors? So they have ChIP-seq data. They know which genes GATA4 and TBX5 regulate. And so we can do an in silico deletion experiment and see, are the affected genes enriched for the target genes versus housekeeping genes? And this figure shows on the left random housekeeping genes, and on the right are the target genes. So we do see, yes, statistically, the effect in silico mimics what's happening experimentally with ChIP-seq. You can go on and on with this, looking just at knockdowns and see, does this affect what people see when they do single-cell RNA-seq or bulk RNA-seq? The SGPT authors showed that knocking down DAD1, you see this predicted gene expression. It basically mimics or correlates what's going on experimentally. And this is our own paper. We have our own method that shows that basically the directionality of the gene expression matches experimentally. Not always the magnitude, but there is sort of the right directionality. So this looks promising that we might be able to do these things computationally. Why would we want to simulate perturbation when we have CRISPR, we have things like perturb-seq? Well, I mean, these can be conducted genome-wide on all cell types in a particular tissue, which is just a massive amount of computations that just can't be done experimentally. And we can do this even on, say, post-mortem brain tissue. We certainly can't do CRISPR screens on post-mortem brain tissue across all cell types, so it's really access to hypotheses that you couldn't get to experimentally. But I do want to have a cautionary note that this sort of training these pre-trained models on healthy tissues and getting single-cell RNA-seq, those models may not have seen all the possible perturbation states you get when you actually do something like knock down tubulin genes. I mean, that's not something you're going to find in nature, but you could do it in a perturbation experiment. So what you'd really like is to train these models on perturbation data. The problem is there isn't that much of it in the world. There's only a few genome-wide perturb-seq data sets out there. So for now, we'll have to live with these caveats around training on healthy cells. So back to this kind of building a case for a target. I think this is where LLMs can be interesting and increase efficiency for researchers. They can have sort of rapid access to target phenotype observations in literature. If the model is trained right, you can even get something about causality, like target X inhibits target Y, for example, depending on how you do the LLM. And these can be great just for annotating gene lists. Say you just did an in silico perturbation experiment and you have 100 genes of interest. How are you going to analyze those things? You can do gene set enrichment, but here's a way you can quickly basically know if a target is connected to an indication through querying literature rapidly. So you can see every big tech company has their own version of a LLM now, and then smaller companies are beginning to use RAGs and sort of refine these for different needs like surveying literature. So we have an interest in this particular commercial product causally. It is kind of a graph of scientific literature that has a co-pilot on top of it, so you can literally just ask it text-based questions, and it's turning those into a graph query and then giving you back a response. I think it's pretty interesting because you can do things like type a question of interest to you. What are the targets for Parkinson's with genetics evidence, for example? And it will pull back all the relevant publications for that question and then click a button for an in-depth analysis, and it essentially writes a one-page review article on the 20 most relevant papers. So that's an incredible time savings if you were to sit down and read and process 20 papers versus have an LLM write you a review article. So this is an efficiency gain, not necessarily an access to new hypotheses, but I think what will be great with LLMs is this concept of agentic AI. So LLMs that actually understand workflows and can call other code to do things for you. So imagine if you could speak to a co-pilot or a chat bot and say, I have this RNA-seq data set, go and batch correct it, go and do differential expression on it, tell me what pathways. So this sort of language interface that you might have to a student, if a chat bot can have access to different agents that can do different tasks, then this could be an interesting way to do basically research in the future. So you'll hear a lot more about agentic AI from other companies. Okay, so that was kind of target ID. Now to think a little bit about small molecule design, what can Gen AI do for us there? Making molecules is an interesting problem because of the massive size of chemical space. I think this is probably an underestimate, 10 to the 60. So there's a lot of chemistry out there that could be done, but you do want to squeeze this into particular parameter spaces to fit the solubility, the permeability, lipophilicity clearance, these kinds of properties on top of on-target activity and lack of off-target activity. So it's quite a needle in a haystack to make the right small molecule. So wouldn't it be better, instead of just searching for it, to design it? I mean, that's really the way to access this massive chemical space is de novo design, not screening. So the principle with generative chemistry is you have a latent chemical space, and you may start with a non-optimal molecule, like a seed molecule, and you would sample that latent space, but only in the property spaces of interest. So maybe you want to improve activity, you want to improve log P, and then decode the structures that actually are in the right property space. This is called multi-objective optimization. So I think generative chemistry is a way to quickly get to chemical structures that have the right properties and hopefully the right activity. So we've done some exploration in this. We've collaborated with Microsoft Research a bit, have some algorithms. One interesting thing is There are different ways to represent a molecule. You can use the SMILE string, you can use a graph representation of a molecule. You can be liberal or conservative about the generation. So this algorithm called molar is actually very conservative. It preserves the core scaffold and then just tries to optimize the R groups around it. And chemists love this because that's how they think normally, is just modify the R groups during lead optimization. But there are generative methods that are much more dramatic in what they suggest. And one of the ones I want to mention is something we call PQ-SAR to compound. Because another way to represent a molecule could be by its biological fingerprint. So what is a molecule done against, say, 14,000 prior diverse biological assays? If you had that value, it'd be an incredible fingerprint describing the molecule. Now we haven't, although we have over 14,000 assays we've ever run in 20 years, we haven't run every molecule through every assay, of course. But what you could do is build a QSAR model for every assay you've ever run. Take a novel compound, maybe it's brand new to the archive, run it through that model and basically compute a profile QSAR. So it's this very rich biological fingerprint, and it's great for similarity searching and scaffold hopping, but it can also be used for generative chemistry. So you can use that PQSAR profile as the condition on which to generate structures. And we have this method called Jaeger that does that. So given a biological profile you want to match, give me the chemical structure that could fit that biological profile. And we had a test case starting with a seed molecule that was an anti-malarial, and we computed its biological fingerprint, and then we went to our pre-trained model and said, give me more compounds that would match that biological fingerprint, and we made 2 of these molecules, compound 1 and 2, and they were both active. And you can see that they had very low similarity to the seed molecule that we input and very low chemical similarity to really anything in the training set. So this particular approach is great for scaffold hopping in the process of molecule generation. But what if we don't have any seed molecules? What if we're starting from scratch? We know we're interested in a target like, say, WDR5, which interacts with MYC, the oncogenic transcription factor, and we just have pocket information. So classically, we would do things like virtual screening, molecular docking, but what if we could just design a molecule for that pocket? So this is kind of a 3D generative chemistry approach. We have our own method called Pocket Crafter based on the pocket to mole algorithm that's published, and we put it head-to-head against a large-scale million compound diversity screen, which had a hit rate of 0.025%, and then we designed about 2,000 molecules and found sort of the closest match without synthesizing 2,000 molecules. 7 of those were hits, so we had hits. We had about a 14-fold enrichment over our diversity set. These are some of the structures that had activity. They weren't necessarily more potent than the best high-throughput screening hits, but one interesting thing is these molecules we designed accessed residues around the pocket that nothing else we found actually touched according to crystal structures. So it was sort of interesting in its creativity in finding new ways to interact with the target. Once you've got these ligands designed, you really want to know where they're binding. Hopefully in the pocket you design them for, but in ligand-based approaches, you may not always know. So these are kind of this new field of co-folding proteins with small molecules. And AlphaFold 3 was sort of like the latest, greatest, best version. It is still proprietary, at least the aspect of folding with small molecules. Others are trying, so David Baker's Rosetta Fold All Atom is another attempt at this. So interesting, the 2 on the left are the Nobel Prize winners in physics this year, Hassabis, John Jumper, and David Baker. And then there are some open source versions. There's something called UMOL that we found from this year. Chai-1 just came out a couple weeks ago, so these are happening. In parallel, there's a whole initiative called OpenFold, and OpenFold has already released a version kind of like AlphaFold 2, but they're pushing it to co-fold with small molecules. The interesting thing about co-folding is you might find allosteric pockets because you're not actually designating a pocket, you're just saying fold this protein sequence with this small molecule. So I think this, you can imagine the future is generative chemistry combined with co-folding in a really good scoring function. So the last thing I want to mention is like AI-enabled assays. I mentioned how you could take a simple bright field image. You can do that in a high school biology class. Take a bright field microscopy image. It's simple, but what we do normally is we stain features of cells. In this case, we're looking at myelin, the protein sheath that's on the outside of neurons and protects neurons. And sometimes you have these diseases of demyelination. We were looking for drugs that could increase myelination on neurons. The protocol was kind of arduous, doing this staining. The problem is when you want to stain a neuron, you have to fix it, in which case you've killed the cell just to take the image. What would be great is to do kinetic imaging where you're not killing the cells. So we trained a model on a joint distribution of bright field images and myelin-stained images. And what we found then is we didn't even need to do the stain. We could just take a bright field and do a virtual stain using a generative imaging model. And this was super powerful, and it's quite accurate. It matches well with the real myelin stain versus the predicted stain. The project team loved this because then they didn't have to kill their neurons. They could just keep taking bright field images over time and get actually kinetic parameters of remyelination. And then finally, you might ask, it's not just myelin stain. Of course, we could do something like cell painting, the Broad cell painting protocol that has 5 or 6 dyes. We can actually simulate a cell painting image given a bright field image, which is a huge time savings because it's not such a trivial thing to do. We and others, also the Broad has shown you can do that. But what if we went really crazy and said, can you predict a cell image for a compound without even taking an image of the molecule whatsoever? Could we use its chemical structure and predict what it would look like if you treated cells? I think when my team suggested this, I said, there's not enough data to do that. There's not enough chemical diversity coupled to, say, cell painting images to even think about doing that. And it's true, it doesn't work, but what did work is not using chemical structure, using that biological fingerprint I described earlier called PQSAR. So basically, we can take a compound, compute its virtual activity across 14,000 legacy assays, and then we've pre-trained a model on PQSAR fingerprints versus cell painting images. And this worked really well. This just came out in bioRxiv like this month, October 12th. You can see here's 3 example compounds. The image on the left is the real image in cell painting. Image in the middle is our prediction using chemical structure. It looks terrible, looks nothing like the real image. And on the right is the prediction using PQSAR to image generation. So we call this PDIF for profile diffusion. So that's intriguing. How extensible is this? Can we just do cell painting now on chemical structures without actually doing the experiment? So my final thought here is thinking about AI and what it's going to do for us, and thinking about it in the context of automation and what that's done for society. I'm going to cite yet another Nobel Prize winner from this year, Daron from MIT, who wrote Power and Progress. So he talked about productivity revolutions in the past. When Ford Motor Company created automation, it was really the fact that it was combined with new products and new tasks and new ways of working that made it powerful. Not that they could just automate the same cars that everybody else was making. And so I'm thinking about what is the parallel with AI here. I mean, automation AI could replace chemists. It could replace biologists in the future. That's not what I see what's happening. In those cases, wealth tends to concentrate in the hands of the tech owners that own the technology. You could see that happening with AI. The few companies that are really big in AI could start becoming really wealthy and owning this space. But more pertinent to this conference is, can AI actually just create new tasks, new ways of working, make us more creative as opposed to replacing workers? So that's what I hope is the role of open innovation in AI. And finally, you can imagine traditional drug discovery being more efficient and more creative with AI. That's the ambition. I would say we have examples of becoming more efficient but less creative, and that would be like combinatorial libraries in the past, where we had fewer hypotheses but more compounds. I'd say we're here with AI right now. Basically, we are getting more creative, those examples I showed you, but it's not really made drug discovery more efficient yet, so this is obviously the ambition. Can we get more efficient and more creative? Test fewer targets, make fewer molecules, and I think we'll get there through method maturity and access through open innovation, success on projects, and then adoption. And hopefully we move to that right quadrant. And I thank you very much for your time.